About the male. Let's talk about the male reproductive system. Again, we've done it in lab, so this should be super familiar. We're going to focus on the stuff that's different or that we haven't discussed in lab, okay? So the male reproductive system mainly is gonads. What are gonads? Ooh, darn it. What are gonads? The main reproductive organ, right? Your testes in the male. Then we have a system of ducts, because the gonads are where those sperm come from. That's the male reproductive cell, the sperm, right? And then it goes through a series of ducts in order to get it to the outside of the body, starting with the epididymis, which is right next to the testes, all the way to the urethra, which is going outside the body. And then we also have some accessory sex glands. What do? Well, the seminal vesicles, the prostate gland, and the bulbourethral glands all produce a secretion that either is a part of semen or prepares the urethra for semen to go through it. Okay? It's okay because we're going to break it down. Don't freak out just yet. Okay. And then we have supporting structures like the scrotum and the penis. The scrotum is meant to hold the testes outside of the body. Why do you think the testes are outside of the body? Because body temperature is too high for sperm production. They will die in that heat. And this is super cool because I don't usually talk about that, but I talked about it earlier this week, and one of my students said, you know, that's really interesting because she works in a clinic, and they had a patient or a couple that were infertile. They're trying to have a child, and they couldn't. And she said, we've done all these tests, and we could never find anything wrong with either one of them. We didn't know what was wrong. And then we found out that they were using a hot tub every night. They had a hot tub, and they would go sit in the hot tub every night. And when they told them, okay, you need to stop the hot tub use, they actually were able to conceive a child a few months later. Yes. High temperatures will kill the process of sperm production. Just for your knowledge, okay? Now all the girls are like, we're going to stick them in saunas before they come see us. Okay, and then the penis, which is really just a delivery system. The penis is only meant to get sperm closer to the oocyte. Do y'all notice how she left right before we did the male reproduction? I'm going to go give it to her at lunch. I'm like, you stayed for the boring stuff, then you leave on the good stuff? She's like, I'm out. Okay, we're going to talk about these in detail. Well, I had to finish the endocrine. I didn't want to spend any more time on endocrine, so I rushed through it. But I know she's going to be like, you talk too fast. I'm like, okay, we were trying to hurry. Whatever. Okay. Was I talking way too fast? I always talk fast. That was weird. I know. And she was complaining about the cold and the light reflecting, and I was like, oh. Oh, she's just pissed that the screen in there isn't as nice as this old one. It doesn't matter. She's always counting down anyways. She's got the days counted down till she retires. She's like, she told me yesterday, she said 3 years, 71 days, and 5 hours. I'm like, okay. But thank you. Y'all did great. You had great questions. Awesome. Okay, I love that. Okay, so the scrotum is really just that sac that holds the testes. No big deal, nothing special. The testes, these are the big boss. This is where sperm are created. Yeah, this is what it's all about. Without the testes, there's no reproduction. So 2 of them, they are oval and paired, one on the left, one on the right. Guess what? You know that the tunica albuginea, I hope some of you didn't know because I've graded your tests already. The tunica albuginea covers the testes, right? That connective tissue that protects them. Outside of the tunica albuginea is a tunica vaginalis. And that one is one that actually coats the testes and also the inner wall of the scrotum. So you have 2 layers to it, a parietal layer on the scrotum and a visceral layer on the testes. No big deal. Again, not going to worry about it too much. So inside of these testes are seminiferous tubules. That is where those sperm cells are becoming developed into mature sperm, okay? And then in between those seminiferous tubules, those interstitial cells, remember those? Those are the ones that are producing testosterone. So testosterone is going to aid in maturing those sperm. I'm trying to rush through this so I can stop and pause on the important stuff, which is about to come, okay? This is not here, but this is a quick look at the inside of a testes. Notice that the tunica actually divides the inside into little sections, and each one of these sections will have 1 to 3 seminiferous tubules, okay? So in these tubules is where sperm cells are developing, okay? And then when they have a head, an acrosome, and a tail, they're going to be pushed out into the rete testes right here, and that's where they kind of sit and chill for a little bit. Then they go on to the epididymis. This is the epididymis here. Okay, and we're going to learn that the epididymis is actually a major part of this, because even though those sperm were developed here, they're stored in the epididymis. They are also activated in the epididymis, meaning even though they have a head, an acrosome, and a flagellum, they're still lacking some stuff. They still don't have the tools they need to move that flagellum. They don't have all the enzymes they need for their acrosome. All of that happens here in the epididymis. That's how important the epididymis is. So the epididymis takes these sperm cells that, you know, have a head, acrosome, flagellum, harbors them, and also helps complete their maturation by activating them, allowing them to swim, giving them everything they need, bless you, and then stores them. They can be stored inside of the epididymis for 4 months. If they're not ejaculated after those 4 months or when they begin to die, they're going to be reabsorbed in the epididymis. So the epididymis is sort of like kind of the end all of everything, right? Like, yeah, they're coming from here, but the epididymis has a big job to do. Okay? All right. So here's what it looks like. Gross anatomy. I wanted to show you this because some of you had trouble with this on the test. This is the testes. This is the epididymis. You see how it just kind of cups right over, sort of hugs on that testes. And yes, they are open onto each other because the rete testes has to send all that sperm into the epididymis. Okay, and then from the epididymis, they're going to go up through the vas, right? And then we'll talk about that. And then they're going to connect into the urethra from the vas. Okay. Spermatic cord is the protective covering. Like if I were to look at what's inside of this spermatic cord, I'm going to say there's a vas deferens, there's an artery, there's a vein, there's lymphatics, there's a nerve. All of that is wrapped in spermatic cord. Make sense? Okay. Okay. What time does this class end? Oh, we got time. Okay, good. Okay, so I want to talk about, and this is probably what's going to take the most time on the male because after that, everything's easy. But I want to talk about spermatogenesis. And spermatogenesis is the process of taking a stem cell and evolving it to become a sperm. Or spermatid, sorry, spermatid. Okay, let's see how we do that. So, right here, this is the outer edge of this tubule. And on the inside of this tubule, on that outer edge, there are stem cells for sperm, and they are called spermatogonia. Do you see these large cells here? Those are spermatogonia. Okay, at puberty, this begins, and then it does not stop until this male dies. So at puberty, at the onset of puberty, those spermatogonia will begin cloning themselves. So I have a stem cell called a spermatogonia. It is going to make a copy of itself. So now I have 2 spermatogonia, 2 daughter cells that are identical. Notice that spermatogonia are 2N. Do you know what 2N means? It means it is a diploid cell. What does that mean? It means it has 46 chromosomes. Okay? And we know by now that that's double the amount you need for a sex cell. You only want half that, right? Okay. So the spermatogonia makes a copy. It has, now you have two identical daughter cells, each have 46 chromosomes inside of them. One of those cells is going to remain at the edge here on that basement membrane, and it is going to be a stem cell again, right? It's going to be an extra cell. The other daughter cell decides that she's not staying home. She's going to run away to Vegas and be a stripper. Okay? Yeah. So the cell that decides, the spermatogonia that's running away to Vegas to be a stripper, I so wanted her to be here for this, runs away to be a stripper. She'd expect it. The one that decides to run away to Vegas, okay, once it removes itself from that area, it starts to undergo changes. If you were to run away now, leave your parents' home, I know most of you are not in your parents' home, but if you were to run away now from your family on your way to Vegas with nothing, by the time you get to Vegas, you're going to be a different person. There's going to be stuff that has happened to you. Things will change. And that's what happens to this cell. Things change on the inside of the cell so that now it is called a primary spermatocyte. Notice a primary spermatocyte still has 46 chromosomes. And what made it a primary spermatocyte, it moved away from the basement membrane and said, hey, I'm going to Vegas to be a stripper. I ain't got nothing to do with these stem cells here. I'm not doing it. Okay? So that primary spermatocyte is the one that is going to go on to give us at the end those sperm cells, but I want to do that on the next slide. Okay, I left that animation in there for you anyways. Okay, so right here at this picture, this was your spermatogonia or spermatogonium, that first stem cell that made two daughter cells or two copies of itself. One of them stays as a stem cell right there. The other one runs away from home, becomes your primary spermatocyte. Going to Vegas, things changed on the inside. Still have all my chromosomes, but I'm not a spermatogonia anymore. I'm a primary spermatocyte, okay? So that primary spermatocyte is going to undergo meiosis the first time, meiosis 1. When it does that, it is basically just cutting it, oops, sorry, this must be in half, cutting itself in half. Okay, it's basically just cutting itself in half. When it cuts itself in half, those chromosomes will be split, oops, sorry, split in, none of that happened, huh? Will be split in half. When you split the chromosomes, you end up with 23 here, and 23 there. That's what we call an N, or a haploid cell. Okay? See what happened? So that primary spermatocyte cut itself in half, gave you two secondary spermatocytes. Each one of them took a half of that DNA. So now I have two secondary spermatocytes that are each haploid, meaning they have 23 chromosomes. Now each one of those spermatocytes is going to undergo meiosis. Again, so this is the second meiosis that has happened. Right? Interpret that any way you want. So this is meiosis, but this is the meiosis in which we're not, what happened is, and this may be a little confusing, but watch the animation if it does confuse you. This N right here, this actually had a copy of its same chromosomes, right? Does that make sense? Okay. So it splits those copies and gives you two daughter cells. Okay? The other secondary spermatocyte gives you these two daughter cells. So these two here belong to this one, and these two here belong to that one. Okay? That gives me 4 spermatozoa. And it is those spermatozoa that are going to go on and continue maturation to be 4 individual sperm cells. Yes? Maybe? Okay, let's erase it and look over it again. Tell me what the problem is. I'm feeling a problem. No? We're good? Spermatozoa. Well, these are spermatids. The spermatozoa are when they begin to develop more. Okay, let's. I confused you. I confused you.
Those are still 23 chromosomes each?
These?
No.
These are 23 each. They are all haploid. These are haploid spermatids. Okay? Now look, I want to point something out. Going from a spermatogonium to right here, spermatids, which is these four right here. From there to there, that is the process of spermatogenesis. Okay? Once you have your four spermatids, you're no longer splitting cells, you're not dividing chromosomes, you're not making copies of anything, right? You've got four spermatids. The process of taking those four spermatids and giving you four viable sperm is called spermogenesis. Okay? Does that make sense? So spermatogenesis is from the start. Starting with a stem cell, you end up with four spermatids. That is the dividing of the cells, the copying of the chromosomes, splitting up things. Once you have four spermatids, you no longer need to divide anything. They have everything they need. They just need to sit in the oven a little longer. So when you're cooking those spermatids into sperm, that is called spermogenesis. So it's pretty easy coasting after this. Parts of a sperm, really, you've got the head. Inside the head, the nucleus has all of that DNA, the genetic material. Those acrosome on top of the head is what has those enzymes to help it break through the outer shell of the egg. Then you have a neck that connects it to the middle piece. The middle piece has all of the mitochondria. That's important because there's lots of ATP required by a sperm in order for it to swim around. And then you've got your tail, which is made up of a principal piece and an end piece. Hormones controlling the function of the testes. So at puberty, gonadotropin releasing hormone comes from the hypothalamus. Remember, your hypothalamus is secreting either a releasing hormone or inhibiting hormone. So at puberty, it's releasing the gonadotropin releasing hormone. That goes to the pituitary, the anterior pituitary, and tells it, hey, we need you to release our gonadotropins, which are FSH and LH. FSH is your follicle stimulating hormone. LH is the luteinizing hormone. What do? Well, follicle stimulating hormone is going to go to the testes and stimulate maturation of sperm or spermatogenesis. LH is going to go to the interstitial cells that are in the testes and ask them to begin secreting testosterone. Okay? Now, that testosterone that you're going to get is going to help things like not just maturation of sperm, but also give you the male pattern of development. You're going to have male sexual characteristics showing. Sex organs will be growing, and then anabolism. Anabolism is protein synthesis, meaning, yes, I hate to admit it, but this is why males, genetic males with testosterone, will have more dense bones and larger bodies and muscles, because it's all proteins. They're able, you know, if I was lifting weights and I had a dude with me that lifted the same exact weight as I did at the same time, his muscles will be bigger, simply because he's a male and he has that testosterone that helps him with that protein synthesis. Okay, and my back hurts too. All right, so here it is in short. What does testosterone and dihydrotestosterone do? Prenatal development. Remember that in the womb, everybody starts the same, and it's the hormones that control what this embryo becomes, whether it's a male or a female. It's like taking the same piece of Play-Doh and either shaping it into a female sex organ or shaping it into male sex organs. Okay. It also helps with development of the male sexual characteristics, things like thick facial hair, deep voices, sexual function, stimulation of anabolism, which is that protein synthesis, and all of that works by a negative feedback system. Most of our hormones are controlled by negative feedback. The one that isn't, sorry, I just had a flashback. I thought we already did this. We really haven't done this yet? Y'all just let me talk? I love it. Okay, we did it. Thank you. Moving on. Okay. Have we done this? Did we talk about the epididymis? Y'all are just killing me. Okay, so that was sperm and testosterone in the testes. Then when they are fully developed, like they have a head, an axon, a tail, they go on to the epididymis. Did we talk about the function of the epididymis? Because it's a big job. Okay, because the epididymis is the one that's going to store the sperm for up to 4 months. It's going to activate them, allow them to swim, give them the enzymes, and then if they stay in there for 4 months and aren't released, or if they come into the epididymis and they're not healthy, they can be reabsorbed. Okay, we talked about the vas deferens. Yeah? And how it opens up here with a common duct called the ejaculatory duct along with the seminal vesicle. We did. In the urethra at the level of the prostate, and the prostate also has ducts that secretes prostatic secretions into the urethra. Right now what I'm seeing is a mixture of yes, we did it, and oh man, she must have done it, but I was dozed off or I was daydreaming. I don't remember a word of this. Okay, gotcha. We'll do it in short. So the epididymis holds on to the sperm until there is sexual arousal. That's when the sperm get moved into the vas deferens, okay? So they're living, they're housed, they're boarded in the epididymis. They get shoved into the vas deferens when that male is aroused. They're getting ready to be ejaculated. So they'll move into the vas deferens. The vas deferens opens up into a duct called the ejaculatory duct along with the seminal vesicles, okay? So now your semen is made up of the sperm and a little bit of fluid coming from the vas deferens and the seminal fluid coming from the seminal vesicle. And that opens up into the urethra at the level of the prostate. The prostate also has ducts that it can put its secretions through into the urethra. So all of that combined is going to be semen and ejaculated during ejaculation. The spermatic cord, this is our protective covering. And here, what are you going to find in the spermatic cord? The vas deferens, along with an arteries, veins, nerve, and lymphatics. Nothing special. Okay, the ejaculatory ducts, that's what I was just talking about. That's going to be, we didn't do this, did we? Oh, we didn't. Okay, okay. So the ejaculatory ducts are the junction of a duct coming from the vas deferens and the seminal vesicle. Ductus deferens, seminal vesicle. Those two together give you an ejaculatory duct. It opens on into the urethra. The urethra takes it from there all the way out of the body. So the urethra is that common duct for the urinary and the reproductive system in a male. Okay, now we have some accessory glands. We have seminal vesicles. Those are the ones that produce seminal fluid. Remember, that joins the vas deferens in the ejaculatory duct. What is this fluid, seminal fluid that we call? It is alkaline. It's viscous, meaning it's kind of mucousy and thick. It has fructose, prostaglandins, and clotting proteins. Whew, I know, right? The prostate also contributes to that, and it is that gland that's wrapped around the urethra. It gives you a milky, slightly acidic fluid that contains citric acid, proteoleic enzymes, acid phosphatase, and seminal plasmin. All of the stuff coming from these two guys right here, these two glands, those are all things that are meant to aid sperm, either by providing nutrition or by helping break down mucus on the pathway. Everything is meant to help the sperm get to its target, which is that little egg that it wants to fertilize, okay? And then we have the bulbourethral glands. The bulbourethral glands will be secreting a clear mucus that is meant to clear the pathway of that urethra in case there's any bit of urine left over that might be acidic. The bulbourethral glands will secrete their secretion through, clear it out, and also provide some lubrication. So the bulbourethral glands will actually secrete the secretion in the beginning. This is when arousal begins, right? These two are ejaculation. This is what people call pre-cum. I'm sorry for the term, but this is during arousal. I can't spell it either, but makes sense? So we got one type of gland that's preparing for the arrival of the sperm by clearing everything out and adding some lubrication. And then we have these two that are actually giving the fluid that the sperm are bathed in order to keep them healthy and alive and able to swim until they get to the egg. Okay? So semen, average volume of semen per ejaculate is 2.5 to 5 milliliters. And you can have up to 150 million per sperm. If you have 5 milliliters of ejaculate or semen, and each milliliter has 150 million, that means that I could have up to 750 million sperm per ejaculation. Wow, it's a lot. Yeah, that's why I keep telling y'all, oh man, it's nothing. Like sperm are so common. There's so many of them. And it only takes one little tiny microscopic sperm to get you in trouble. Just saying. All right, the pH is 7.2 to 7.7. The penis, nothing special. Guess what? The penis is only meant to be a device that helps bring the sperm closer to the egg. Okay, it is just a delivery system, helps extend it out of the body and into the female so that it can be closer to the egg so they can survive that swim much better. It does have 3 masses, the 2 corpora cavernosa and 1 corpus spongiosum. Hopefully you got that right on the test. If you didn't, you're gonna have to learn it for lecture. And then the glans penis is the head, which is at birth covered by a foreskin or a prepuce. And here it is in a real section. I think it's kind of cool because you can see all of the holes in the corpus cavernosa. See how all of that is waiting to be engorged with blood. And then your corpus spongiosum right here with the urethra inside of it has a little bit more of that soft tissue so it doesn't engorge as much. Pretty cool, huh? That's all a penis is. Yeah. All right. An erection. An erection is brought about by the parasympathetic nervous system. But guess what? Ejaculation is brought about by the sympathetic system. So you have to have a working parasympathetic system that upon arousal, whether it's mechanical or some other type of sensory sensation that initiates this erection, that's the parasympathetic system. It kicks in, allows those blood vessels to dilate, blood pools into the corpora, causing the penis to be erect. But in order to get to ejaculation, where the sperm are actually expelled out of the penis or out of the body, you do have to have sympathetic stimulation for that. And then there are some muscles that aid in the ejection of sperm, and that is the bulbospongiosis, the ischiocavernous, and the superficial transverse perineal muscle. Those will also contract during ejaculation in order to help push the sperm out of the way. Or in, I don't know, not out of the way. Okay, I left this in here because it's super cool. Look at this. This is talking about erection or ejaculation. This is the connection of our brain or males' brains to their penis, right? Get this, mechanical stimulation. So these are mechanoreceptors that are in the penis that can sense mechanical stimulation. That sensory neuron takes that sensory impulse to the spinal cord and it reflexes right back through parasympathetic onto those arterioles, allowing them to dilate, allowing the penis to be erect. Okay? Notice this is a reflex arc. It didn't even require any brain up here, right? This happened without the brain's involvement. Remember, your sympathetic and your parasympathetic are the autonomic nervous system. We always called it the automatic nervous system. Okay, so this is an automatic response here. Other things can play into this. Let's go look at the head here. So we have erotic sights, smells, thoughts, erotic sounds. All of these are sensory inputs that can also have the same effect. So if it comes from the brain with some type of sensation of the special senses, whether it's something they thought or smelled or heard, that can also send a neuron down to the parasympathetic and cause the same type of erection, which is kind of super cool. So I'm just showing you this because it's neat. And please understand that, you know how we joke about men having two heads, right? It is kind of true, y'all. This here, right here, it's on its own, okay? That's not something they can really control. So be kind. Be kind to those simple folk.